My name is Deborah Wall-Palmer. I'm a Marie Curie Fellow at Naturalis. I have a background in marine biology, but also in micropaleontology. That's the fossils of tiny creatures. We're preparing for the AMT-27 research cruise. And right now we're preparing a lab before the ship sets sail, and we have to make sure everything is secured really well. These barrels will be used to prepare some water for experiments, and we will fill each one with filtered seawater, and then we'll change the pH of that water by bubbling through different concentrations of carbon dioxide gas, so that we can subject gastropods to these different pH waters, which represent water predicted for the future and also water from the past, to see if the effects of ocean changes affect their shell growth.
So you're now back from the expedition. How did it go?
The expedition was really successful for the plankton team. We collected lots of specimens and carried out lots of experiments in a long transect through the North and South Atlantic Ocean. And the results of some of those experiments have recently been published. We investigated the effects of changing ocean chemistry on a species of swimming snail that lives in the South Atlantic Ocean, Atlantidae. It's a small snail of about 3.5 millimeters across. It has a flattened disc-shaped shell and it has a foot modified into a swimming fin. As an adult, they are carnivorous, feeding on other zooplankton, that's animal plankton, but as juveniles, they feed on algae, and we use this juvenile stage in our experiments because they're thought to be the most vulnerable stage to ocean changes. So we got lots of specimens using a plankton net, and we subjected them to 3 different ocean chemistry conditions, one for the past around the mid-1960s, one for the present, and one for the future around the year 2050. And we looked at how their shell growth responded to the different ocean chemistry conditions.
What did you find out?
So we measured the shell in a few different ways. First, we used calcine staining to measure how long the piece of shell grown during the experiment was. And then we used micro-CT scanning to work out the shell thickness and the shell volume grown during the experiments. And we used transcriptomes, that's how the genes were expressed by the snails in the 3 ocean chemistry conditions. We found that in the past conditions, the snails grew a longer piece of shell and a higher volume of shell compared to the present day. However, we also found that the snails grown under the future conditions also grew a longer piece of shell compared to the present day. And the expressed genes also showed that there were unique responses to each treatment. In some cases, the expressed genes were upregulated or downregulated in a linear way with, for example, decreasing pH. But in other cases, it was more like a U-shape where the genes were up or down regulated in the future and the past compared to the present. These results are not straightforward, but we can conclude that shell growth in Atlantidae was more stressful in the future conditions, and it will be negatively impacted in the future.
Why is this important?
So the shells are really important for Atlantids. They use them for protection, but they also use them for swimming. They actually use their shell as a secondary swimming fin. And under the future conditions, they grew a longer piece of shell, but this was really stressful, and it's unlikely that they would be able to maintain this level of shell growth under future conditions. This could mean that they grow a weak shell, and this will impact their ability to hunt as adults. Atlantids are selective predators, and so they're really important for the ecosystem. So losing them would have far-reaching effects for the ecosystem in which they live.